And yeah, yes, my field of research is mostly genomics, metagenomics, and microbial ecology from the sequencing point of view. So I'm leading the bioinformatics service in FISABIO. It's a foundation for public health research in Valencia. But the focus here is to show you a kind of new way to represent data, and for instance, to see, at least I will try to transmit to you this, the feeling that is really easy and just mail to Kaveh and he's able to do magic things with his tools. So at least this is my main objective for this talk. Yeah, I will speak about metagenomics. You know, metagenomics is a way to access the environmental property, the genetic properties that we can find directly from the organisms that are living in a given environment. We can approach the environment using, for example, this one, metataxonomy. Metataxonomy, or people call it also meta-barcoding or barcoding. So it's just a way to get, using a PCR reaction, a tag, a label for all the organisms that are present in a given environment. It doesn't matter if it is seawater, soil, fecal sample for gut microbiome, etc. So we had these labels, and then we count how much we have for every kind, and use database to assign a taxonomic annotation to every single pieces. On the other side, there is metagenomics. That is not the previous one. Metagenomics extracting DNA from the environment, breaking the cells, so we lose the identity of every single cell, and releasing this DNA, we fragment, we share it apart randomly and sequence everything. So from this sequencing, so from these random pieces here, we try to assemble them using our bioinformatics magic stuff, and finally trying to annotate them. So on top of this assembly, we identify the sequence. From the sequence, we identify the gene, and from the gene, we identify the function. So finally, it's an inference of the function that may be carried out in a given environment, potentially carried out in a given environment. I say potentially because this is genomics, is not transcriptomics. So we know what the bacteria can do, but not what they are really doing. Well, the final aim is always to identify pathways like this. This is a very simple one where we have enzymes that are making reaction from one point to the other one, producing or acting on top of a given metabolite. And so one after the other, finally, we reconstruct the complete metabolic network of an organism. I say an organism because it's the simplest, at least if you can see here simplicity. This is not simple at all. And here, for example, this is the metabolic network that we can build using only the Escherichia coli genome, or Serratia symbiotica, Carsonella, etc. These are, in order, genomes of bacteria that are reduced genome size, so reduced complexity. But still, just one organism is very, very complex stuff to work on. Imagine when we are working not with only one organism, but if we were working with a metagenomic sample. This meta is for metapopulation, so it is a complex population made by maybe thousands of different organisms together, and also important is to understand that the frequency of this is completely different. There are overabundant organisms, and some of them that are very, very, very tiny represented. And so from all this, we get the DNA, we annotate, and we can build a big network like that. These networks, as I said at the beginning, we don't have the membrane, so we don't know who is doing what. So we have everything at the same time. One approach is, for example, to reduce the network to some compressed stuff that we call a minimum metabolic block. So imagine we have an input here, an output here, so we can compress everything is inside it, just one small block. And then we can easily make things kind of easier with this metabolic block. This approach was called the MetaDAG. It's a paper, it's a work that is still in progress by my colleague Mariana Reyes and other two researchers from the University of Islas Baleares. And we are working together trying to apply this system and reducing the information to make it more easy to work with. Still, when we go to a metagenomic sample, imagine this completely chaotic network is very, very difficult to work with that. Also because the network is not fixed. It's something that moves. We can change the topology of the network. So we really don't have a clear vision of what is happening in a so much complex system. Yeah, my work, okay, I come from CNR in Messina 20 years ago, then I moved to Valencia where I started working with very noisy or very, very bad human gut microbiome sample, you can imagine, and, but it works also for that. In gut microbiome, for example, we are interested to understand the change in gut microbiome during the aging of the people. Or, for example, another interesting stuff is understanding the effect of diet. This is a kind of milestone for me. It's a paper from a group of De Filippo in Italy, and they were studying children from Burkina Faso and compared it with children from Western, our children that are mostly eating hamburger against the African one that are mostly eating cereals. And we see how the bacteria can change from one group to the other. The good one are the other. The Western one are the worst microbiome you can find. And also a very hot topic is understanding if enterotypes exist. So we have blood types, but also we are interested to know if we really have a group of people that share the same or similar microbiome, and then if this can be used as an estimator for risk factors for health. So with all of this in mind and the complexity offered by metagenomics approach, there was one day, a couple of, I don't know, 6 months ago, we started working together with Cave. Laura said me, you have to know Cave, you have to work with him because you can do beautiful things with him. And then I was thinking to how to represent this in a graphical point of view. And after a couple of discussions, we reached a kind of reference cube, a reference block that is to avoid all the topologies of the metabolic of the metabolic reaction. So my original idea was just this piece of PowerPoint that I sent to Cave, where I can represent pathways in layers, and then I can connect vertically those enzymes that are making the same function in two or more different pathways. In such a way, I fix all the pieces of the puzzle, and then like a cube of LED, I have to turn on and turn off every single enzyme depending on the results I have for a metagenomic experiment. At least this was my starting point. Then I sent this to Cave, and Cave started doing this. And here there is Laura saying, wow, look what Cave did with your data. And this is a beautiful system because we have this three-dimensional cube that is a metabolomic genomic cube, so a metacube. And at the moment I finished my presentation at this point because I was really excited when I saw this picture, and then after I don't want to make any spoiler, but Cave is going to give us a demonstration about that. And finally, just a few words. I would like to use this moment also to promote a conference we organized in Spain in November 2025. It's the One Health Pangenome. Is a conference where we are going to speak about comparative genomics so looking at the pangenome really, and but the idea is to look at this comparative analysis from the healthy point of view, public health point of view, and then moving to the agri-food system, and then veterinary and soil, and coming back from the environment to try to close the circle of genetic evolution among all these three compartments. Here there is the website. I think it's already active. Here is our mail if somebody wants to start being in touch. Still, we don't have a program, but we are working just in this day on that. Just fix this on your calendar. Just finally, I want to thank you everybody for the attention, and for sure I want to remember Merced Llabrès y Pere Palmer from University of Balearic Island in Spain, Laura Cave for sure, and Mariana that is actively working with me on this project, and you for your attention.